Hi everyone. On behalf of QuTEM, I'd like to wish you all a warm welcome to today's webinar, and we really appreciate that you can spend some time with us today. My name is Cara Poon, and I'm a senior scientist at QuTEM, and I'll be your host today as well. Today's webinar will involve a presentation followed by a Q&A session. So if you have any questions during the presentation, please feel free to write your questions in the chat box to the right. Your questions will also be private, so please don't hesitate to send in your questions. Only our admins will be able to view them. We'll also try our best to answer all of your questions today, but if we do run out of time, we'll get in touch with you afterwards by email and answer your questions there. Finally, after the webinar, we'll also share a link to a recording of the webinar, so please don't feel any pressure to take any notes as well. And now it's my pleasure to introduce our presenter for today, Rickard Nordström, COO of QuTEM. Rickard has been with us since 2012, ever since our days as Vernova Bioanalytics, before we were acquired last year. And Rickard will be talking about TEM analysis of AAVs for quality control. So with that, I'd like to invite Rickard to take over the screen.
Okay, thank you very much, Cara. It's a huge pleasure to have this webinar, and I'm so happy to see that so many are joining. So today, in this webinar, I will talk a lot about the transmission electron microscopy analysis that we do in QuTEM, and specifically what we can do for you when all of you who are working with adeno-associated viruses, or AAVs. So just a very brief background on who we are. As Cara mentioned, QuTEM acquired Vernova Bioanalytics, a TEM company that has been in Stockholm since 2007. And our focus is really on providing transmission electron microscopy services for the pharma industry and for the biotech industry. And since this is so important, since this is our niche, GMP is very important. And that's why we also have aimed and got a GMP certification from the Swedish Medical Products Agency. The GMP certification means that we have qualified facilities, validated equipment, validated methods, and qualified personnel to ensure that everything that we deliver to you in form of data is traceable and reliable. We also have a team of 30 employees with all expertise that we need in electron microscopy, virology, image analysis, and software development, and quality assurance. We have also developed our own software called GridSea. Since it's fairly or comparably easy to acquire images in a transmission electron microscope, it is, however, quite difficult to extract information and make that into valuable and quantifiable metrics. And that's why we have made GridSea. So GridSea is actually the core of what we're doing. It's an image analysis software, but it's also record-keeping software, and it's fully integrated with transmission electron microscopes that we have. It's also, of course, full. So while we are located in Sweden, in Stockholm, Sweden, we help clients all over the world, both in North America, in Europe, and we also have quite a few clients in Asia as well. Most of our clients work with drug and gene delivery system, such as AAVs, but also other systems. We're doing quite a lot of analysis of vaccines, and every now and then we also do other type of analysis on other type of nanoparticles. And our clients are in all different stages. Since we're GMP certified and our methods and analysis can be used for batch release, there are a lot of our clients are in clinical or commercial phase, but we do also have quite a few who are in early R&D and in R&D phase, because it's quite important to set the methods as early as possible, as you know. And I will talk a lot about AAVs and QuTEM today, but we do analysis of a plethora of different particles. We also do quite a lot of different analysis, but the ones I'm going to focus on today is the three uppermost, the vector content analysis. So basically determining if the AAV particles are full, empty, or have intermediate forms of genome inside. I will tell you a bit more about our methods for genome length determination for AAVs, and then I have a few slides on aggregation of AAV particles. But before I start talking about the analysis that we do, I would just give you a very brief overview of how gene therapy in general works. So common for all gene therapy vectors is that it's targeting a dysfunctional gene. So a patient has typically an inherited genetical defect that causes a dysfunctional protein that is causing this disease. And this is what you try to target. So instead of taking care of the symptoms, you're actually trying to replace the dysfunctional gene and thereby restoring full functionality. So here is just a schematics over a cell with a dysfunctional protein. You could see these as AAV vectors. So all of them, or most of them, are carrying a gene, a DNA string that encodes for a functional protein. And typically this is an engineered gene. What you would like to have is all of the particles should be full. I will come back to this later on. So this product is then administered to the patient intravenously. And depending on the type of virus or the type of vector, there are different routes for cell entry. But commonly for all of them is that the genetic material is then exposed to the interior of the cell, and the normal cellular machinery will take care of the transcription and translation of this genetic material into the functional proteins. So why is quality control important then? Well, and this is typically what we can provide you with using TEM. Because what you would like to have is the full vectors with the correct genetic material, because these are the ones that encodes the functional proteins and cure disease. The empty vectors, they do not have any biological activity. They do not carry any gene. So they are effectively an impurity, and the only thing they could do is actually trigger an immune response. More problematic forms could be partially filled or overfilled vectors, and this could be a plethora of different subspecies. So either it could be particles that have packed just a fraction of the genome, or they have packed something from the host cell, as an example. And in worst case, those could, of course, translate other proteins and give various adverse effects. So let me tell you a bit more about the quality control analysis that we do in QuTEM. So this is the typical workflow after you have placed an order on one of our services. So the first step is that you send the samples to us for our analysis. And one of the good things with TEM is that you need very little material. For one analysis run, you only need 3 microliters. So typically we ask you for 25 microliters. But and if it's really a scarce product for you, we can actually do with less than that. So these are shipped to our GMP laboratory here in Stockholm, and then we store them in the correct temperature, and everything that we have here, all the different temperatures, the ultra freezers and fridges are of course monitored. The next step is the sample preparation. So we do perform two different sub-techniques called negative stain TEM or NSTEM or QuTEM. I will tell you a bit more of these about these later on. And we do the preparation in our biosafety 2 laboratory. And being biosafety level 2 means that we can, in principle, take care of any of your AAV products and also various different other viruses. The next step is the TEM imaging. So we are using two Thermo Fisher microscopes. The one behind here is actually our biggest workhorse, a Glacius microscope. It's a 200 kilovolt microscope that can take 12 samples in one go. And immediately when the images are acquired, they are directly imported into our proprietary software and the analysis starts. Next step is the image analysis. And here is a typical example of AAVs that has been acquired in QuTEM. And already here you can start to see the dark disc-like particles. That's fully filled particle, and then the ring-like is actually an empty particle. So GridC will automatically detect the particles and then extract the quantitative metrics, for example, the internal density. And this is what I would like to talk to you about more today. So the next step is then reporting and delivery, and the reporting is also built into GridC. So all the analytical data is compiled into these analytical reports. And then we have a step of independent review before this is delivered to you together with all the images that were acquired. And typically, this whole analysis takes less than 10 business days. We also have an option if you're in a hurry called AAV Express, which is just 4 business days before you have the results. And we are, of course, always available should you have any questions before, during, or after the analysis. All right, so a bit more technical then. The two techniques that I talked about, the NSTEM or negative stain TEM and QuTEM, they are in principle the same, use the same type of equipment. The biggest difference is that negative stain is in room temperature and you stabilize and stain the samples with a heavy metal stain. In QuTEM, you don't do basically any tempering. The only thing you could do is the dilution of the samples, but there are no additives or anything. So the sample is rapid immersion frozen using a freezing robot, and the specimen remains in its close to native state. In negative stain, you get enhancement of the surface structures of the particles. So it's super good if you want to look at the integrity of the particles, if you want to identify small structures, debris, and such. While QuTEM is very much like an X-ray image that you get from the hospital. You will look inside the particles as good, and making it the method of choice if you, for example, want to look at the presence of the genome inside the particles. Some example images, you have AAVs and adenoviruses, and you can see here on your left-hand side in the NSTEM, you can clearly see all the details. However, the adeno-associated virus, if they are intact, they all look like bright discs. While if you look inside the particles, like you do here in the QuTEM image of the same AAV sample, you see that some of them are full and some of them are empty. And we know that there are companies out there that do AAV analysis or full empty analysis using negative stain TEM, but that is really not the method of choice. All right, so a bit more about the semantics when it comes to filled, empty, and full AAV particles, because well, empty, it's easy to describe. An empty particle doesn't contain any genome at all. Anything else Whether it's the correct genome or not could be considered filled or full even. However, there's typically only one of these that represents the correct form, the gene of interest, which means that any other form that is filled but not with the right genome is unwanted. So here they are regarded as partially filled. In some cases, as I described before, the particles could pack more material than you intended, and these are actually overfilled, like in this schematics. So in short, the relative values are really not sufficient to describe an AAV product. It's not sufficient to say that they are full or empty, but you need to know more about the genetic length to be able to describe these subspecies. So in the last couple of years, we have developed a method to do exactly this, to be able to better describe the various filled forms that are present in a sample. And we do that by actually determining the genome length of each of the particles. In order to do that, you need to take care of a lot of different parameters. In TEM, there is a lot of things that you need to take into consideration to make one image comparable to another one. So these are just a couple of examples just to show you what we do image processing wise. So here on the left-hand side, what you see is an uncorrected QuTEM image where you have a bit of background contrast gradient. So it's slightly darker in the bottom than in the top. So what we do is first is that we do a noise reduction and we do a frequency filtering to flatten the image. So we subtract this low frequency information and the outcome is a fully flat image. Since there are differences in electron dose and differences in ice thickness, we also need to make a correction of the transmittance. So we do a normalization to do that. And most importantly, we need to be able to pick the particles and pick the background. This is done by our software. You also need to be aware that AAVs are icosahedral, so depending on the orientation, they could either end up in the projection looking as a in a hexagonal form, or you see all 10 vertices. So what we do is that we segment out the background, like in this picture, in the middle picture here, and we also segment out the particle shell. And we use this information for the normalization and to determine the internal density of each of the particles. So a typical analysis data or analysis run could look something like this. So this is the raw data from, I think this is about 3,000 particles or a bit less. So here on the y-axis, you have the number of particles. On the x-axis, you have the internal density. And you can already now see that it's not a lot of noise, but you clearly have 3 different peaks here. So what we do is that we make a Gaussian fit of these. And you can see that it fits fairly well, actually. And if you look at some examples of particles that are in each of these classes, you see that it's actually the low density particles are empty, and then you see the high density particles are full, and then you have this intermediate form. So the next step is that we wanted to describe these better. So we'll spend quite a lot of time in building a reference database where we have well-characterized AAV particles with known genome length. So typically they have been sequenced, and by other means we've been able to verify the length of the genome. And then we have calculated the internal density of each of these, and thereby building a reference curve. This reference curve can then effectively be applied to any data to determine the length of the genome for each of these subspecies. So in this case, we have the empty ones are 0 kb, the intermediate ones 2.5, and the most full ones 3.8 kb. So a typical readout from our analysis, so this is actually 2 diagrams that we have in the report, can look like this, depending on what you order. So this is a typical readout for the genome length. You get the raw data and you get the Gaussian fit curves. And we also give you the genome length of each of the subspecies. And here on the right-hand side, we have replaced the diagrams, or the diagrams, the curves with bars, showing the relative percentage of each of these classes. So the empty class constitutes 37% of the population, and the filled class 62%. I know that quite a few of you have already used our services, so I would just like to spend a few minutes just to go through some of the details in the analysis data that we provide. So in this diagram, we have the possibility of actually having several subspecies, like in this particular case here. And the farther to the right, the longer the genome is, but we don't provide the exact genome length in this diagram. So both of these are filled, but they constitute 2 different types of 2 different lengths of genome. So this is the full one, 65%, and this is partially full. This is 4%. And if we have a well-characterized and known product, we can customize the category names. So in this particular case, this is actually the N-terminal of the N-terminal construct containing just a GFP. And this is a truncated form of the same genome. Looking at the genome length determination curves, it's actually the peak value that corresponds to the length of the genome. And the width of the curve is rather related to all the accumulated measurement errors that comes from the technique itself, because there are differences. Even though we do normalizations, there will always be small differences. And we try to minimize this and make the curves as sharp as possible. But if the curve look like this, it shouldn't be regarded as if you have varying lengths of genomes in this one. Should you have that, what you will see is a skewness of the curve. So in this case, if it's skewed towards 0 kb, it could indicate that you have presence of partial truncation of the genome. Quite commonly, we also see a plateau like this between the peaks or even outside the peaks, which may indicate that you have presence of polynucleotides that comes from the host cell. And these do not form a single peak, but they rather are of varying lengths. So you probably wonder, what is the performance of the method? How well does it work? So what we have done also to make it easier for you to take this analysis into use is that we have done a platform validation on our end. So would you like to use the method and ensure that it's validated for your product and fit for intended use? We can easily set up a small study just to verify that the validated platform work for your product. And this is the factual results from our validation. So when it comes to the vector content determination, so whether the particles are full, empty, intermediate, in the repeatability testing, we had a standard deviation of 1%, and in intermediate precision, it was even lower, so it was less than 1%. We also had a sensitivity of only 50 particles. So we only need 50 particles picked to determine that we do have a subspecies in the product. When it comes to genome length, the repeatability had a standard deviation of 150 bases, and in the intermediate position was 110 bases. Also in this case, we had a sensitivity of 50 particles. And typically the resolution was plus minus 75 bases. So it's quite a neat technique to determine the length of the genome as well, and to characterize different subspecies that you have in your products. So finally, before I conclude this presentation, I would just like to show you a few slides regarding some of the other analysis that we can do for AAVs. And in this case, it's an aggregation analysis. So what you look at here is actually a negative stain image of AAVs. This analysis can also be performed using QuTEM. The good thing with using negative stain is that you can see, you get a bit more information regarding the integrity of the particles. It's also possible to see genetic material, should you have that, or other proteins that might be present in the sample. So typically what we do in GridSeq is that we segment out the particles that are individual, and we segment out the aggregates, and then we would also provide you with the area or the size of these aggregates, looking like this. And it's fully possible to do this also in cryo. What we have seen is that TEM is really particularly powerful when it comes to identifying small particle aggregates, and that is aggregates smaller than 500 nanometers, although it's possible to also identify larger aggregates. But particularly good if you are concerned about small clusters with 1, 2, 3 particles, as an example. And in the analytical reports, we will give you the percentage of individual and aggregated particles. We can give you the size of the aggregates and so forth. So just to conclude this presentation today, transmission electron microscopy is a powerful technique to determine the integrity of the genome in AAV vectors and to detect and measure small particle aggregates. And thank you so much for your time, and I'm ready to answer any of your questions.
Thanks a lot, Rickard, for your presentation on AAV analysis using TEM with QuTEM. So now we'll have just a very short Q&A session. If you haven't sent in your questions yet, I invite you to do so now in the chat box to the right. So now we'll begin taking some questions. So the first question we have actually is, how many samples can be prepared and then shipped to our lab for cryoanalysis?
How many samples?
Yeah.
I think we have a very high capacity. Actually, we could easily run 50 samples a week without much problem. So I think we can run whatever is needed.
Mm-hmm. Absolutely. All right, so our next question is also, how do we address aggregation issues? And I think this question actually came in with respect to the QuTEM question. And then by the same token, I see there's some other questions here. Do aggregates and impurities affect then the vector content analysis, for instance?
Mm-hmm. So on your last question, we haven't seen any impact actually, since it's as long as the particles are not overlapping. If you start to have very large 3AD aggregates and we cannot segment out single particles, then it's not possible to do the vector content determination. But when you have these 2D clusters, it is fully possible, and it doesn't really impact.
Mm-hmm. Perfect. Okay, so the next question we also have is, what are the key advantages of cryo-TEM over a negative stain TEM?
So I would say that, first of all, you can see the interior of the particles. You don't add any stain. We also see that for some specimen, the preparation of negative stain actually requires the sample to be dried, and that could actually give you a bit of movement of the particles on the grid before they are stabilized and fixed, and that could actually induce a bit of aggregation. So overall, I would say that the QuTEM keeps the specimen in its native state. Negative stain is super good to see small debris and the integrity of the particles.
Absolutely, would agree. So now we also have a question about how do we address the debris and the ice formation in cryo-TEM? I know you touched on it a little bit earlier, but maybe we can expand a little bit on that.
In the QuTEM, well, the debris typically don't impact all that much. If you think the ice formation in terms of hexagonal ice that comes from the atmosphere, we do have systems built in GridC again to filter out any such ice contaminations, as we call them, but they are basically ice crystals that are deposited on top of the grid.
Yeah, and those are quite normal aspects of the QuTEM technique as well. Yeah. So now we have a couple more questions. How do we differentiate double-stranded DNA inside an AAV, and also impurities?
So impurities in terms of proteins, we cannot really do at this at the resolution that we've chosen to have. When it comes to single-stranded or double-stranded DNA, we have done some testing. We would like to do even more. I would say that maybe that's not the main use of this technique, though. What we have seen so far is that typically it doesn't really matter for the density whether the genome is single-stranded or double-stranded. It still has the same density, so to say.
So yeah, we would say that neither really affected the genome length analysis either. Is that correct? Yeah.
Yeah, correct.
Yeah. Then when it comes to is peak determination fully automated, or does an expert interpret the results?
It is fully automated, and we always have actually two steps of verification. So first we have our qualified image analysts that are manually verifying all the data, even all the particles and how they are picked. And then we have a separate step with an independent reviewer that goes through everything once again. So that's part of the GMP delivery that we have. So it is automated, but it's also checked by a qualified operator.
Yeah. And then we have one more question here. Can you tell us a little bit about the standard deviation from aggregation measurements?
Oh, I wish I could, but we're doing the analysis right now, right?
Yes.
So I think we'll have to come back to that actually to give you the exact performance and numerical value of this. But we have seen correlations, and we have done a lot of testing when it comes to known samples, which are more and less aggregated. But this is something that we are investigating and plan to validate in the near future.
Okay, so I think we're coming to the end then to the questions. So I would like to say a big thank you to everyone for joining today, and also for all your questions. Before we go though, I'd also like to share some extra materials with you. So you should now start seeing a few handouts coming up on the right-hand side. And just give me a moment while I share them all with you. Okay. We'll also keep the webinar session open for a couple more minutes more, so that you have time to download them as well. After this webinar as well, we'll send you a link by email so that you have access to a recording of today's webinar. And if you have any further questions, please don't hesitate to get in touch with us at info@QuTEM.com. And finally, on behalf of the QuTEM team, I would like to say a really big thank you for joining us online today. It's been a pleasure to go through our AAV characterization analysis with you, and we look forward to getting in touch with you as well. So have a great day. Thank you and goodbye.